Hi there, in this video we are going to discuss another hallmark of the cancer, which is evasion of the cell death. We know that when the DNA of a cell is damaged, or when there is a accumulation of the mutation inside the cell, what happens? Cell is subjected to the apoptosis or the programmed cell death. For example, this was a cell here and it was nucleus and it got mutated, and there is stress and it will lead to the programmed cell death or apoptosis of the cell. However, in cancer cells, there is restriction to apoptosis, and cancer cells don't die despite of having too much mutations. So, why it happens? Cancer cells, basically, we know that cancer cells are facing a lot of stress, internal stress, which is in the form of mutation, as well as when cancer cells grow, their demand for the blood supply increases. However, the blood supply is not too much to finish their or to sustain their needs. So they are also having the reduced blood supply. Okay, reduced blood supply, as well as they are also suffering from hypoxia. And moreover, when chemotherapy started, these drugs also induce stress to the cell, and it can lead to their death by apoptosis. Okay, so chemotherapeutic drugs. So these are different factors which lead to the stressed internal environment of the cell, and it can lead to their apoptosis. And apoptosis in cancer cell mostly occurs by mitochondrial or intrinsic pathway. So this is the principal pathway which is involved in this entire scenario, by which they can die, and by which they try to evade, by modifying which they try to evade the apoptosis. Other pathway of the cell death is extrinsic pathway, which was a fast pathway. We previously had studied fast. There is fast ligand which bind to fast receptor and lead to the cellular death. However, we are not concerned with that pathway. We will be dealing with intrinsic pathway. So what happens in intrinsic pathway? Basically, we should know some protein factors before we dive into the details of this pathway. There is a protein family which is BCL2 protein family, and this BCL2 protein family has multiple proteins. Some of them are pro-apoptotic and promote apoptosis, and some are anti-apoptotic. Pro-apoptotic members are the proteins which lead to apoptosis, include BAK-BAC and BAX-BAX. These two proteins are pro-apoptotic proteins, and anti-apoptotic proteins include BCL2, BCLXL, and MCL1. These are anti-apoptotic proteins. And another set of proteins is BH3 only proteins. This is the name, BH3 only proteins, which are basically sensors, and you can call them sensors of cellular stress, and these include BAD and BAD bad, BID bid, and PUMA PUMA. So these are basically three sets of proteins which regulate the intrinsic pathway. So when there is a balance in favor of pro-apoptotic members, when they become active, there will be apoptosis. When anti-apoptotic members are active, or the balance is towards anti-apoptotic members, there will be no apoptosis. So what should be the basic purpose of cancer cell here, or basic goal of cancer cell here, to avoid apoptosis? They should basically lead to one way or other. They want to inactivate pro-apoptotic proteins. They want to inactivate pro-apoptotic proteins, and they want to upregulate anti-apoptotic proteins. These BH3 protein class 3, they are also inclined to apoptosis. They sense the stress, and they also shift the balance towards pro-apoptotic members. Okay, so they are basically, also you can call them pro-apoptotic. So these proteins are pro-apoptotic and these are anti-apoptotic, and no cancer cell want to upregulate these anti-apoptotic members, and they want to inactivate pro-apoptotic members in order to sustain their survival and avoid apoptosis. So how they can do it? One way is upregulation of these protein or anti-apoptotic protein, and yes, they do upregulation. BCL2 is principally upregulated in many types of cancers, and this BCL2 upregulation prevents them from apoptosis. However, in order to avoid apoptosis, they also inactivate these proteins. And how they do it? We know that there is protein P53, the product of gene TP53, and this protein is basically have a strong linkage with PUMA, PUMA, which favors the apoptosis. So what happens in cancer cells? P53 is mutated. Okay, when there is a mutation in this pathway, When it gets mutated, the function of PUMA is also restricted. And the function of PUMA is restricted, there is not a shift in balance towards pro-apoptosis or apoptosis. Okay, so in this way, by mutation of p53, there is also a balance that is shifted towards anti-apoptosis, and apoptosis does not occur. I hope so I'm not complicating the things. Let me summarize. uh, Cancer cells are prone to death by intrinsic pathway or mitochondrial pathway. This pathway is dependent on BCL2 protein family, and this BCL2 protein family has three subset. One is three principal subset which we will discuss. One is pro-apoptotic member, which include BAC and BACs, anti-apoptotic member which are BCL2, BCLXL and MCL1, and a third class of protein which is BH3 only protein, which include BAD, BID and PUMA. So this third class of protein is also inclined to apoptosis. Okay, so you can call them pro-apoptotic also. So this entire set you can call it pro-apoptotic, and this set is anti-apoptotic. So cancer cell want to avoid apoptosis. In order to avoid apoptosis, what do? They upregulate these anti-apoptotic members. For example, BCL2 is upregulated in the follicular lymphoma. In follicular lymphoma, there is translocation 14 to 18, in which BCL2 gene is translocated to immunoglobulin chain, and it's upregulated. As a result, there is an anti-apoptotic response and cell don't die, and there is follicular lymphoma, which we will study in the blood system. So in one way, they are upregulating the anti-apoptotic members. In other side, they want to inactivate them. So how they inactivate them? In cancer, there is also mutation of TP53, and this TP53 mutation leads to the down regulation of the PUMA. And we know that this PUMA was favoring the apoptosis. When it is down regulated, there is no favoring of apoptosis, and there is also some degree of down regulation of their function, and cell attain the mutations and don't die. Under normal condition, cell die after accumulating some mutation. However, when there is down regulation of PUMA, cell sustain survival even after accumulating the mutation, which would normally lead to the cellular death. So in this way, the cancer cell evade the apoptosis. So basically, how apoptosis occurred? When there is pro-apoptotic signal, this BAC and BACs pathway, what they do? They lead to the pore formation in the mitochondria, and this pore formation lead to the leakage of cytochrome C, and the cytochrome C activate caspases, which lead to the cellular death, which we have studied in the mechanisms of apoptosis. So we will see the diagram here in the book also for our own understanding here. See here what was happening. This cell is stressed by reactive oxygen species, radiation, chemicals, deficiency of growth factor and so on. So there was a DNA damage, and in DNA damage, when there is DNA damage, P353 response occurred, PUMA was upregulated, and it led to the upregulation of pro-apoptotic member including BAC and BACs. They induced a hole in mitochondria, cytochrome C leaked out, and caspase 9 and caspase 3 got activated, and there was apoptosis. However, what cancer cell do? In cancer cell there is loss of function of p53. It can be either by mutation in p53 or due to overexpression of MDM2. We know that MDM2 holds p53 and prevents its function. So either TP53 is itself mutated, or there is amplification of MDM2. Both these lead to the loss of p53 function. As a result, there is a loss of functionality of BACs and BACs, and there is accumulation of mutation, and there is no apoptosis. Moreover, anti-apoptotic member BCL2, BCLXL and MCL are also upregulated, and in this way cell avoids the apoptosis. So it is basically what's going on here. And to summarize it, you can read this summary here. That summary basic understanding, and this summary will be enough for you. Okay, so next we move on. So we know that how we can prevent this from going on and lead to the death of cancer cell. It can be if there is you can say if there is amplification of MDM2, which lead to the loss of function of TP53. You can synthesize drugs which inhibit this MDM2, and when this MDM2 will be inhibited, P53 function will be restored, and moreover the BH3 proteins, BH3 like proteins can be formulated which perform in their way, and they also activate the pro-apoptotic members. So in this way you can target the cancer cell towards apoptosis. However, this is in clinical trials and requires extensive research. Our next topic is next cancer hallmark is limitless replicative potential. We know that in normal cells, supposedly this is a chromosome here, and these chromosome ends, normally chromosome ends have some portion of DNA sequence and proteins which are called telomeres, and these telomeres are basically specific DNA and proteins which are structured which are attached at chromosome ends. And what they do, they basically prevent the DNA repair mechanisms. They prevent DNA repair mechanism from identifying the ends as double strand breaks. Okay, if there are no telomere, DNA repair mechanism become active and they recognize these ends as double strand breaks, and they can lead to the arrest of cell cycle and senescence. Okay, so what telomeres are doing, they are basically preventing the senescence or the DNA repair response. So what happens when cell continue to divide again and again, this telomere get shortened. Supposedly here was its length this, and in the next division its size will go down. Supposedly in next division the telomeres have shortened, and in the next division they will shorten more and more, and as a result a stage will come in which telomere length is too much shortened that the cell is not capable of dividing more, and there is loss of cell division. How this loss of cell division is occurring? Because after losing the telomeres, DNA repair mechanism become active and they recognize these end as DNA double strand breaks, and DNA double strand breaks is basically, you can say, a mutation or fault in DNA that lead to the senescence or the arresting of the cellular growth. However, cancer cells require some mechanism by which they can overcome this you can say tragedy of telomere shortening, because it is a tragedy for cancer cell if their telomeres become short, they would not be able to divide. So what they do to avoid this? Firstly, what happens in cells, there is a, you can say, chromosomal binding pathway becomes active in which chromosomal ends, you can say this is a chromosome and this is other chromosome, and here is telomere, and these telomeres have shortened too much. What happens? Non-homologous chromosome join, and in anaphase these chromosomes moved are attracted towards opposite poles, and there is, you can say, abnormal joining of chromosome, and there is abnormal mitosis which lead to mitotic crisis, and this abnormal mitosis, too much mutation accumulated can ultimately lead to the cell death or massive, you can say, apoptosis. So first there was telomere shortening, and after telomere shortening, if cell tries to divide, a tumor cell tries to divide, there is mitotic crisis in which non-homologous chromosome join and they are attracted towards opposite pole, and it lead to massive mutations and it lead to the massive apoptosis. So what other way they adopt to avoid this, they activate this telomerase enzyme, and when their telomerase enzyme become active, this telomerase enzyme synthesize back these telomeres, and as a result telomeres length is maintained and cancer cell don't die off or don't stop their growth. Also you can understand it by diagram from the book here, so you can appreciate this is a cell here and there is inactive telomere, and when telomerase is not active, cell can maximally live to 50 to 70 division, and after that telomeres are shortened, and when telomeres are shortened, cell is subjected to the senescent state, or you can say it is no longer dividing, and it is sent here by p53 pathway. However, if there is a defect in p53, so what will happen? Non-homologous chromosomal joining pathway will be activated, and what will happen as a result of this, chromosome will join. You can see this chromosome, these two chromosomes have joined here and these two have joined here, and these chromosomes are being pulled to opposite side in anaphase, and it lead to the many mutation inside the genetic setup of cell, and this increased mutation can lead to the mitotic catastrophe and can lead to the cellular death. However, in order to avoid all this scenario, what cancer cell adopt? They activate the telomerase enzyme. This telomerase enzyme synthesizes back the telomers, and telomere length is restored, and the cell continue to divide. In this way, cancer cell attain the limitless replicative potential, and they are not subjected to the arresting of the cell cycle. So it was all about today's lecture. Thank you.